So good day. I'm happy to be able to present as part of the Good Oral Candidates 101 course the topic BCS, the Biopharmaceutics Classification System, and BDDCS, the Biopharmaceutics Drug Disposition Classification System, tools to facilitate efficiency in drug discovery and development. On this slide, I've listed the learning objectives that I hope you will be able to take away from this presentation. Define the difference in terms of both objectives and methodologies between BCS and BDDCS. There's a lot of overlap, but we want to know what the differences are. What steps would you take to determine the BDDCS class of a new molecular entity? Describe the relevance of enzymes, uptake, and efflux transporters on the disposition of drugs within each BDDCS class. What is the rationale for intestinal membrane permeability being the determinant of a major route of drug elimination in humans, which when you first encounter this question seems really strange that intestinal membrane permeability would tell you about metabolism. Why will BDDCS Class 1 compounds yield central or brain effects for drugs that are efficient substrates of brain efflux transporters? And describe how would you estimate the potential for a new molecular entity to be primarily excreted unchanged in the bile of humans? So in 1995, Professor Gordon Amidon and colleagues, including FDA scientists, proposed the Biopharmaceutics Classification System, designated BCS. The core idea in the BCS is that an in vitro transport model centrally embracing permeability and solubility with qualifications related to pH and dissolution may qualify for a waiver of in vivo bioequivalence studies, which in fact led to the 2000 FDA guidance on BCS and biowaivers. The objective of the BCS is to predict in vivo performance of drug products from in vitro measurements of permeability and solubility. However, when we looked at the BCS, we thought there could be other aspects of BCS and that it could serve the interests of the earliest stages of discovery research in predicting the absorption and disposition of new molecular entities, and was the impetus for our lab proposing the Biopharmaceutics Drug Disposition Classification System.